Hi, this afternoon we are going to explore the use of Aqueous Arecanut extract in the treatment of coronaviruses, because we try first using it against influenza virus, which bear resemblance with coronavirus in its symptoms during an infection, in its structural properties and several other properties. So for that we need to really understand what has the work been all about so far. Clinical investigations into the potential medical properties of ARECA, CATECHU, limb. So the first few slides will concentrate on the properties of arecanut extract with respect to antimicrobial, which is nothing but anti-infective property, as well as antioxidant property, which is the ability to scavenge free radicals, that property of the aqueous based arecanut extract. And then we will come down to talking about the proposed work against influenza and coronavirus. The work has not been executed so far, but this is a proposed work plan and the work map. Introduction, preparation of the aqueous arecanut extract. The chapters covered: antimicrobial activity of aqueous arecanut extract, free radical scavenging activity of aqueous arecanut extract, in other words antioxidant activity of the arecanut extract, anti-inflammatory activity of the arecanut extract, anti-ulcerogenic property of aqueous arecanut extract, anti-diabetic activity of arecanut extract, which is anti-hyperglycemic activity of arecanut extract. So how do we prepare the aqueous arecanut extract? Hot water extract of the arecanut was prepared by boiling the nuts around 100 gram in 500 ml distilled water. The extract is subjected to concentration by evaporation, and yield of the extract we figured out was 6.4%. Extract re-suspended in distilled water and diluted to desired concentration. These are the arecanuts, the pictures. This is the right side of the first picture is a nut. It's open and you see we need soft tender nuts. Left is the annulate stem, like fluorescence, and it is an oriental palm, and right is another picture of all that it is all about. The nuts are there, it's in a husk, and the palm is there for you to see. So basically, arecanut palm, tree with annulate stem surrounded by crown of pinnate leaves. Arecanut or betel nut are the nuts or the seeds of the palm, and we are interested in the nuts. Picture 1 that I showed you just now is Areca vestiaria. The wild palm is one of the most beautiful ornamental pinnate palms, comparable only to ceiling wax palm in beauty, which is native to North Sulawesi, the northernmost province of Sulawesi Island. In India we have arecanut palms in the coastal regions from Maharashtra to Kerala and Tamil Nadu. It also grows in the Deccan Plateau, Assam, Meghalaya, West Bengal, and Andaman and Nicobar Islands. However, in the world we find that it is found all over in Sri Lanka, the Malay states, South China, east of India, Philippines Islands, which is Philippines, parts of East Africa including Zanzibar and Tanzania. So we find that you have it in the continents of Asia and Africa. I haven't spoken much about America, but well, there are certain islands off the coast that you find the arecanut palm. Basic constituents, we might be interested in the phytochemical constituent of arecanut: polyphenols, fat, polysaccharides, fiber and protein, alkaloids and tannins. Alkaloids, basically arecolin, arecadin, guaifenin, guaicolin, arecolidin. Polyphenols, we have leucocyanidins, catechin, polymerized leucocyanidins, leucopelargonidin, flavonoids, tannins. Total amount of ashes is 3%. There are macro elements also in the arecanut like calcium, potassium, magnesium, sodium, iron. Poorest source of sodium is arecanut. Trace elements like copper, manganese, nickel, zinc, lead. Okay, they are heavy metals actually, and they are found in trace concentrations. Others such as aluminium, barium, strontium, chromium and silicon were absent in this spice. Now generally arecanut, the betel nut, is taken for chewing with the betel quid. So how exactly it is arecanut plus betel leaf plus lime with or without tobacco? However, tobacco has been implicated in the cause of oral cancer and oral submucous fibrosis. Arecanut could be baked or boiled. When I say the nut is taken, how are we going to chew the nuts? Baked, boiled or raw. Chewing the nut and betel quid alternatively is also another style seen in South Africa. Or chewing only the betel quid, which is arecanut plus betel leaf plus lime with or without tobacco. In northeast part of India, our own country, raw and wet variety of arecanut, betel leaf and variable quantities of slaked lime, which is produced from lime deposits of stones, are generally the combination used for chewing. Kwai is the wet variety, supari is called the dried betel nut. Experimental studies have been done with betel leaves and cardamom, and there's not really any property associated with it. However, betel leaves plus clove, I think in Malayalam it is kariyamba, clove, it shows that oral microorganisms, the oral flora, are suppressed. Betel leaves plus cardamom plus clove, it shows some germicidal effect against bacteria. Traditional pan, as pronounced in India, in Hindi, the language of India, the national language, is betel leaf, lime, arecanut, gulkand, cardamom and clove. So basically spices, it's a spice basically. Let's view arecanut in human health. We know pan masala is a combination of all that I just said. The cardamom, the betel quid, the slaked lime, and we know that it affects the health like mouth, brain, heart, liver, kidney, stomach, general health. Large doses of gutkha can cause cocaine-like state of intoxication. It's a psychoactive substance basically. And symptoms, reddish staining of the gums, teeth, white, painful formation inside the mouth, amnesia, insomnia, fatigue, dizziness. So all this problem, it's absolutely essential that there is a ban on pan masala, but we are not discussing pan masala or gutkha. We are discussing the use of nuts. An extract prepared by boiling the nuts can be useful as a therapeutic agent, can be used as a nutritional supplement, can be used as a functional food. So what we found out, and we would discuss these two chapters just to throw light on the fact that arecanut extract has antiviral properties. It has also antioxidant activities. You know, in many of the diseases, free radicals are produced. The stress is related to free radicals being produced or generated, and scavenging of the free radicals. That property is the antioxidant activity. So arecanut can be exploited for its antimicrobial or anti-infective property against both bacteria and viruses. We know in viral infections there are secondary bacterial infections, so throwing light on the antibacterial activities broaden our understanding as to why it can be used against viral infections, and we are discussing influenza and coronavirus. So the basic properties, the phytochemical constituent of arecanut, all this play a very important role in determining its pharmacological activities. antibacterial, antiviral and antifungal activity of the arecanut extract. So aqueous extract of arecanut is prepared. Strains used: gram-negative test organisms like E. coli, Klebsiella pneumoniae, Salmonella typhimurium. Veterinary isolates as well as human isolates were used. Gram-positive organisms, Streptococcus mutans, we purchased it from IMTech, Institutional Microbial Technology Center in Chandigarh. Streptococcus viridans, strain number 1603 from CMC Weather. So all these gram-positive organisms have been been used for this work as test organism. So we found that 100 ml of nutrient broth was inoculated with the protocol with the strains, incubated at 37 degrees Celsius. So we take sets of tubes. Control is just the nutrient solution with the organism without the extract, a set of control. Another control is just the nutrient broth without the organisms, without the extract. Incubated at 37 degrees Celsius, 0.1 ml of overnight culture were incubated in the presence and absence of escalating concentration of the extract, and we worked on concentration 10 to 100 micrograms. It was incubated at 8 hours in 3 ml of neutral broth. Absorbance of the tubes were read at 530 nanometers because arecanut possesses some kind of color, a chocolate brown color. Immediately after inoculation of bacterial cells, and continued for 8 hours at intervals, different time intervals. Triplicates were done, and each time period, like if we took it for 1 hour, second hour, there were triplicates for the 1 hour, so that we get concordant values and errors can be minimum. Antifungal activity, we worked against mucor, Candida species, Candida albicans, which causes the oral thrush in infants. So it also causes vaginal infection in women. Aspergillus niger, Cladosporium aspergillus flavus. Flavus is known to infect grain stone in chick feed, etc. So the same procedure is found in in the absorbance method was performed using the yeast cells Candida albicans, because it's a unicellular fungus. It can cause turbidity in the broth and grow like a bacteria. This diffusion method was also carried out because the fungus is a mycelial form. It was performed using the mycelium cultures. We also did try to find out whether it had any implication or effect on the aflatoxin production by Aspergillus flavus. So Aspergillus flavus was procured and it was grown in glucose ammonium nitrate medium, incubated, and from the broth 100 microliter was suspended and inoculated to test tubes with glucose ammonium nitrate medium. Extracts from 10 to 250 micrograms per ml were used. Triplicates performed for each concentration. Extraction of aflatoxin was done using modified method for each concentration. Estimation of the toxin concentration carried out, and we can view the intensity of the filter paper in a UV fluorescometer, and we get an idea of the amount of toxins that's produced. So these are results: 10 microgram, 20, 50, 100, like I said, between 10 and 100. Worked against gram-negative human veterinary isolates, gram-negative human isolates, gram-positive human isolates, not taking any gram-positive veterinary isolates. Candida albicans, same experiment: 10 microgram arecanut extract, 20 microgram arecanut extract, 50 microgram arecanut extract, 100 microgram arecanut extract to the left side of the table, that is, and the organism was taken. So 1 hour, 2 hour, 3 hour, 4 hour, 5 hour, 6 hour. Candida albicans, after its growth, we selected a particular, the optical density was determined, and we selected the log phase, early log phase cultures as per 3 ml of medium. So we found inhibition of aflatoxin production by arecanut. We found 35 ppb was the control. Percentage of inhibition absolutely nil. 10 to 50 around 20 ppb, we found that 42.85%. 100 to 250, 5 parts per billion, 85.7%. So we're just trying to establish that arecanut extract has an activity against gram-negative veterinary isolates, gram-positive human isolates. It was fungicidal, it acted against Candida albicans, and there was also a toxin inhibition. Antiviral activity was checked against 2 viruses, the Newcastle disease virus, which is implicated in Arecanut disease strain R2B, and egg drop syndrome virus. It was procured from Manuti. It was a veterinary college which had fowl farm. These viruses were maintained in allantoic fluid of the chick embryonated cultures and embryonated egg in cold storage at a temperature of -70 degrees Celsius. So basically, we need to understand from the procedure, there were around 7 groups set apart, and extract concentration. There were, each group had 5 eggs per set. So one was just a normal eggs. The second was infected with the virus. The second was infected with the virus and treated with the extract. Infected with the virus, treated with the higher dose of the extract. Infected with a higher dose of extract in the superlative degree. And that was done with NDV, Newcastle disease virus, as well as egg drop syndrome virus. And using the candle method, the development of embryo was noted every day. You candle the embryonated eggs. After 5 days of virus and arecanut extract inoculation, after 5 days of the viral growth and arecanut extract inoculation was done, only then all embryos were killed. Hemagglutination titer in the embryonated eggs were determined. Morphology of the embryo and chorioallantoid membrane were noted. So we find there is an antiviral activity as well. The hemagglutination titer, could be seen ranging from this 2048 to 32, 1024 to 56. So some of the references that we could use, as I said, modified Pond's method. We have a reference from the year 1966 used. Then we also have 2004. We have some antiviral activity plans, paper published. So we have established that we have taken Aqueous arecanut extract. We have subjected it to a series of microbiological procedures to find out whether it had anti-infective, that is antimicrobial property against gram-positive, gram-negative isolates from human and veterinary isolates, and whether it had a property against unicellular fungus, Candida albicans, which behaves more like a bacteria. And we also worked against Aspergillus nuca by using the distribution method, and we also tried finding its ability to inhibit the aflatoxin production. And interestingly, we know that aflatoxin works against Streptococcus mutans. Aqueous arecanut extract works against Streptococcus mucus. It causes dental caries. See, we have proved by our antioxidant studies that arecanut extract does have a significant antioxidant potential. Can it scavenge free radicals in vitro? In vivo studies were done earlier by another senior and she said positive results were seen. I have not done any plagiarism by taking her results and showing it here, but in vivo there are beautiful positive results. So what were the battery of antioxidant tests carried out? We did superoxide radical scavenging activity, we did hydroxyl radical scavenging activity, lipid peroxide scavenging activity, DPPH radical scavenging assay, ABTS, it's a diammonium salt radical scavenging assay, then determination of ferric reducing antioxidant power assay, whether ferric is converted to ferrous. So the full form of ABTS is, it's a phenothiazine compound. The full form is 2,2 dash acinobis dash bracket 3 ethyl benzthiazolin 6 sulfuric acid. Results will go one by one. SOD, superoxide dismutase. We found percentage inhibition increases as the concentration of the arecanut increases. Hydroxyl radical scavenging activity, percentage inhibition increases as the concentration increases of the arecanut extract. Blank here is just the butanol and the pyridine in the procedure, not going through the procedure. Lipid peroxidation 15 to 200, and we find percentage inhibition increasing. Blank is only a 3 ml buffer. Control is without the extract. Yeah, test solution without the extract. ABTS concentration 5 to 24, percentage inhibition is seen. FRAP assay concentration between 0.1 microgram per ml of arecanut extract to 5, and you find the absorbance readings increasing. FRAP assay, the intensity of the color, ferrous reduced to ferric. DPPH radical scavenging activity from 0.2 to 10, we find the percentage of DPPH scavenged 15, and then it increases so much of free radical to scavenge, better is the antioxidant activity. Blank is nothing. So effects of arecanut extract on the inhibition of superoxide radical production. We found that by the riboflavin photo reduction method, it increased in a concentration dependent manner, and the concentration needed for 50% scavenging of superoxide was 19 microgram. On the inhibition of hydroxyl radical activity, we found that hydroxyl radical generated by the Fe3 plus ferric ascorbate, vitamin C, DTA, hydrogen peroxide system was found to be inhibited by the arecanut. That means it had positive ability for the inhibition of the hydroxyl radicals. And we found for 50% inhibition, it was found to be 55 micrograms per hour. Lipid peroxide induced by Fe2 plus ascorbate in rat was found to be, and the concentration needed for 50% inhibition was found to be 17.87. Lipid peroxides are found to be reduced. Arecanut extract on DPPH radical scavenging activity. As mentioned, a picryl salt, and the 50% scavenging was seen at 0.4 micrograms per ml. ABTS radical scavenging activity, it was found for the 50% inhibition. It shows in a dose dependent manner. 9.5 microgram per ml was in oxide dismutase. You find dose dependent manner, the graph increases. Percentage inhibition increases as the concentration along the x-axis increases, the y-axis percentage inhibition. Hydroxyl radical scavenging activity also in a dose-dependent manner. As the concentration increases from 10 to 200, we find the percentage inhibition also increasing. But here we find that it kind of remains stable. 76 to 70 is not much of a difference. So there is can increase and then it becomes constant. Here also it increases and remains a constant. Lipid peroxidation, concentrations between 15 to 200 microgram per ml. DPPH radical scavenging activity, concentrations from 15 to 200 micrograms per ml. Percentage of DPPH scavenged increases from 18 to 16. It's a free radical and it has so much of quantity is being scavenged. ABTS assay, we find there's an increase, constancy, and there's a decrease. ABTS assay. FRAP assay, also you find concentration micrograms per ml. With the increase in concentration, you find more the color, more ferric is converted to ferrous. So the color intensity increases. References, we have McCord and Predovich method, the superoxide method. Curcumin is turmeric and it has curcumin. Curcuma longa is turmeric and curcumin is phytochemical constituent, bioactive principle as we could call it. And these are some of the methods. Feral myoglobin ABTS, Alsuraki, Nakara. So there is the need today in modern times to develop natural antiviral. We know acylclovin, sidobudin, these are some of the antiviral drugs available in the market, but we need to have new antiviral agents from natural products. Most of the diseases caused by viral pathogens are self-limiting. Our immune system is able to inhibit the virus and do not require antiviral therapy. However, infections, diseases like AIDS caused by HIV, human immunodeficiency virus, requires drugs like sidobudin, AZT, that is acidotymidine. Available antiviral drugs in clinical settings, generally against respiratory syncytial virus, cytomegalo and other DNA. Three categories broadly of traditional medicines, dietary supplements and functional foods. We could purify the exact, we need to do it. It's not we could, but we need to purify the exact component, the exact phytochemical principle, the exact bioactive component responsible for the antiviral activity in the plant extract. We need to check the efficiency of the same antiviral agent, that is Aqueous Arecanut extract. Could it be used against the severe acute respiratory syndrome virus, coronavirus? So in this regard, we have previous reports carried out on influenza viruses where cell culture and cytotoxicity assay, aqueous extract of the betel nut preparation, freeze-dried extracts were dissolved in maintenance tissue culture medium. The human colonic cancer cells have been used for growing the influenza virus herpes simplex 1 and human influenza A. The cells were grown in monolayer in 5% carbon dioxide and 95% atmosphere at 37 degrees Celsius in the Gypco-VRL medium. It's also called the modified Earls. It has the modified Earls salt with 10% FPS, fetal bovine serum. Gypco-VRL is a company, 10 ml procaine, streptomycin and 5 ml fungizone. We add antibiotics and antifungal agents to prevent the rich tissue culture medium from being infected. Cytotoxicity is tested by growing the monolayer of the cell lines in 96 microtitoplasm. Falcon company produces these and exposed to two-fold serial dilution of the extract. The treated cells were then incubated at 37 degrees Celsius for 3 days, after which they were checked for cytopathological effects, CPE it's called, cytopathogenic effects, and stained with crystal violet to show effects on cellular level. CPE was indicated by clearance of cells in the wells. Clearance of cells in the wells. You may use MTT assay for quantifying the viable cells also. Controls were set consisting of only the cells, and another control was cells with the virus only without the extract. This was proposed, however, it was not executed, that we do toxicity study of plant extract and we do the toxicity study of the standard drug, comparing both by animal studies, mice studies. Since influenza virus and coronavirus show similar disease presentation and spreading nature, we hope we can relate the study to develop a remedy for the current epidemic, which is contained the COVID scare, coronavirus 2019. So study on coronavirus, how well could we carry it out? Since coronavirus cannot be cultivated, cultured, genomic RNA, we have to go for the biotechnology method. The RNA is taken, the cDNA is synthesized, amplification of a certain region of the spike-like protein by PCR, cloning of it into the PFLD plasmid, transformation of E. coli cells and amplification in E. coli DHA alpha, check the inhibitory effect of plant extract through Docky method. So this is what we need to do: genomic RNA, cDNA, the S1 region of the spike by amplification, cloning it into a vector and transforming E. coli cells, checking the inhibitory effects of plant extract with Docky, establish the fact that a particular principle component is responsible for the activity against the sequence.